Hello and welcome everyone to the next seminar in the Research Skills Seminar Series. My name is Sue Scull and I'm responsible for the Research Education Program, and this is one part of that. And we're very much about trying to help busy clinical people, trying to fit research around the margins and upskill them in things they need to know about research. Today is a new seminar for us and very timely. And I think, as you'll hear, many people are really quite unaware of the implications to research of trying to work in the COVID-19 environment. For those of you who are out there offline, please hold your questions to the end. I will be monitoring the chat, so feel free to send questions in and we will raise them at the end of the talk. And we are video linking all around WA, and so that helps with our recording and just keeping things moving. And please fill out your evaluation at the end if you can. It only takes a minute, and we constantly want to try and improve what we're doing. And if you have any comments or feedback, we find that really helpful. So without further ado, I'd like to bring Julie Marsh, who was here just last week also talking about biostatistics. And Julie is a senior research fellow with the Telethon Kids Institute, where she's an expert in adaptive trials methodology, and she's also worked across industry and academia, and she has a wealth of experience in statistical consulting. So please join me in welcoming Julie.
Thank you for those attending today and all those outside the auditorium as well. I'd like to acknowledge the traditional areas of the land, so all the different sites that are calling in today, and pay my respects to their elders, past, present and future. I know you're all busy people, so I'm going to remind you at the beginning and at the end of the topics that I'm going to cover today. So there are existing research guidelines available for the impact of COVID-19. I'm not talking about COVID-19 trials themselves, I'm talking about all the other research which needs to continue during this health emergency. The current guidelines focus particularly on participant safety and also trial governance. We're going to see how long we think COVID-19 is going to be around and impacting our research, and where we may need changes, all these different areas, and where we document all these rationale for the changes and how we decide which strategies to adopt. And finally, how do we address that in publications? And this will also impact how you read publications when you're just sifting through all this research as we start publishing out, in the next 2, 3, 5, 10 years' time. So addressing the existing guidelines. The first slide here is the European Medicine Agency. They have a whole website. I have all the sources at the bottom of the slide there. So if you want to look up any further information, you can go to these websites. So they quickly produced a guidance on the management of clinical trials in particular. And that was updated recently. They're now on version 3 as of the 28th of April. So the areas that they covered in this one was really relating to initiating new trials as well as the ongoing trials. And they really looked at the important thing here is about risk assessment. So that was the focus, and also patient confidentiality and the integrity of the trial moving forward. At the same time, the FDA was developing all of its resources as well, and here's their website. I particularly like the way that COVID-19 gets categorized under counter-terrorism as well as emerging threats to the community. And they also produced a list of guidelines. What was interesting about the FDA one in particular was they produced a list of questions to address for individual trials. And obviously these relate to regulatory trials, but I would hope that we adhere to the same principles of integrity when we do our investigator-led trials as well. I am going to talk more generally about research rather than just focus on clinical trials, but you'll have to excuse me, my background's in trials. It's much easier to come up with examples in those areas. We're also very fortunate that the Child and Adult Health Services has responded rapidly to this situation, and they have a research site as well. Again, the link is at the bottom of the slide here. If you, I'd strongly recommend you all double check and make sure you're adhering to all the policy changes surrounding this as well. And this is largely around the research process and ethics and governance, and addressing things from that point of view. I would assume that most investigators have already taken the urgent actions on their research. So this is really backfilling. How do we maintain how do we do a risk assessment to see if it's worth continuing with our research, and whether it's been fundamentally compromised by the COVID-19 health emergency? And also, if we are continuing, what do we need to take into account? What changes are there in our procedures, in our protocols, in our statistical analysis, and how we report it out the other end? And always keep your eye one step further in terms of when others report out their research findings, what are you looking for in them explaining things in their publications? What will help you trust what they've done as well? So today is all about documentation, all about coming together in your research team with your investigators and discussing how has this impacted us, and how are we going to address this, how are we going to document it. All ideally without looking at any of that data. Okay. The NHMRC has also provided guidelines for us, so there's several different sites listed down here where you can address it. And what I'm really addressing today is the conduct of research related, the emerging situation with COVID-19 is a significant priority. And how do we address that? But how do we also initiate and continue other ongoing and proposed research, which is also critical for the well-being of patients, participants, and communities? So as you can see, the existing guidelines really address the rights, the safety, and the well-being of participants. It's an evolving situation. We need pragmatic solutions. But just drilling down into a slightly more detailed assessment, we need to address the underlying changes in the population that may be recruited to our research protocols. We need to look at changing patterns in our lifestyle and of medical presentations and monitoring, what the impact is of social distancing and mixing, including travel, and that's not just for infectious disease. And also how the COVID-19 situation, as it unfolds, how does that affect adherence or availability of the intervention? So I've had a few people say that their research isn't going to be impacted by COVID-19, which I found amusing. So even if you're doing retrospective data collection, or for example with linked data, reporting it out at the other end, you need to think about how your results are still applicable in the post-COVID-19 world. Okay? For example, the effect of social economic status or potential overcrowding within a household, whether your population you're studying has shifted its baseline characteristics, and whether that's a permanent shift you would anticipate in that target population. So you can still do that retrospective look at the data, but think about how that impacts the post-COVID situation. Yeah? So your discussion is going to get a lot longer when you start reporting out your findings. If you're doing laboratory and animal experiments, there may be obvious changes you've had to make to the interventions anyway. Some of them may not still be possible, with limitations on access to laboratories, or maybe that the laboratory's been turned over to more important testing at this point in time. But there are also small changes that you have to account for. So if your research is ongoing, your own pattern of visiting the lab or taking the observations, or the frequency and timing when the samples are taken may have shifted during the COVID-19 period of time. And also the storage of samples may have been prolonged. So compared to the pre-COVID situation where it was business as usual, the restrictions imposed may have affected things such as sample collection, how the intervention is delivered, or the storage of samples before they're assayed or analysed. Even if you're conducting all of your research online, especially if you're conducting questionnaires, you may see changes due to whether the questionnaire is delivered in person or whether it's done online, and also the qualifications of the individuals who's able to administer that questionnaire, or even if you've moved to it being self-administered as opposed to collected by health professionals. Especially with quality of life, you have to think about underlying changes in lifestyle factors, such as activity, maybe alcohol and smoking consumption, and also stress levels. So I think given some of those considerations, I think there will be few individuals who can safely say that their research will not be impacted by the restrictions imposed on us by COVID-19. So how long is all of this going to be with us? So as of Tuesday this week, this is the Johns Hopkins site, which I believe provides the best source of data, although I'm not quite sure what's happening with Brazil, with their removing it recently from the government reporting, but hopefully that will come back online. And again, there's a website given at the bottom here. So I think we can see that COVID-19 isn't going to go away. So this is a service provided by CITEL, which is a clinical research organisation. And they've been tracking how many clinical trials are active for COVID-19 worldwide, and also the treatments which are being tried as well. Thousands of patients have already gone into a whole variety of different clinical trials. I'm pleased to say a lot of them have been adaptive. And we don't seem to be, we haven't had a real clear winner emerged in terms of the treatment of this disease. And neither have we currently seen any evidence of vaccine efficacy. As it continues with the current restrictions, it will become harder to perform these types of trials to get the individuals recruited in, as we don't have the numbers circulating in the community, which is good at an individual level. However, it's likely this situation is going to continue for years. The vast majority of drugs which have been assessed so far of things which are used for other disease indications, and the other drugs currently in trials from pharmaceutical industries, are often repurposed drugs that they use elsewhere, or something they've taken off the shelf where they haven't been able to find a suitable indication for it previously. So we're really looking for the new wave of antivirals for COVID-19 before we're likely to see any sort of impact in this area. The initial assumption that was made was that we could simply look at periods that were pre, post, and during COVID-19. However, increasingly, that is not going to be the way that we can just stratify the analysis and deal with it that way. Okay. The dates of COVID lockdown are very much dependent on the region. And the extent of which the restrictions are imposed are dependent on whether the case numbers are going up or they're coming down as well. So we're going to have to start collecting information of how was this particular site impacted by COVID-19 between dates, and the idea of that roll-in recruitment. The individual adherence to the lockdown, and whether they choose to make lifestyle changes or other impacts or other risk assessments about leaving the house, going to hospital appointments, or even choosing to enroll in a clinical trial, for example, or other research protocol, they may vary due to social, cultural, or country norms. So a simple stratified analysis will not account for the impact of COVID-19 on our research protocols. I've roughly broke down the issues and how they impact our research objectives into 6 areas. So I called it treatment adherence, but it's the intervention or something which has changed. It's slightly more complicated when you're prospectively looking at observational data, and I won't be covering that in the talk today. But if it's an intervention, there could be disruptions to the availability or the delivery or the access or the administration of that intervention, or different levels of compliance. Some individuals may choose to take holidays and not adhere at all to the therapy during that time, all of which can dilute the treatment effect. The recruitment itself may have slowed down or been suspended, stopped, or even extended at certain sites or even by subgroups. And if you're involved in adaptive trials, that may, in addition, affect the results and when you're able to perform interim analysis and any response adaptive algorithms that you have in place. Protocol violations may increase. You know, for example, individuals being recommended, I mean, particularly in the elderly age groups, when they're very much recommended to remain indoors and not circulate in the community, that also involved not attending clinic visits. So you may find that there are only available data on baseline and, if you're fortunate, at the end of the intervention period. Missing data also comes into that, from disruption to the visit or contact structure, and how to tell the difference between an appointment that would have been missed as opposed to one which is missed due to COVID-19 restrictions. All of these would increase the need for imputation methods to account for missing data and decrease the precision of the statistical model estimates. So, you know, if your outcome is an odds ratio or a change from baseline, you know, we're looking at increased variability around those. There's a potential for bias, particularly in terms of changing participant demographics, or even due to so there's the individuals you're recruiting in, but also differential withdrawals in different subgroups. There's also adherence, and that adherence can even be affected by media coverage. Finally, the confounders themselves. So we may find it necessary to change the trial eligibility. Common ones for those is, do you allow recruitment of COVID-19 positive individuals? Do you allow experimental medications to be taken at the same time? Given that if you're looking at a drug, you may not have even had a chance to look at drug-drug interactions to even be aware of the safety, or whether there are any safety concerns. Recruiting those individuals into your study. So I'm just going to list my way through the different things that you need to address, particularly for the research protocol and the analysis plans. So they're really the two documents that I'm homing in on at the moment. it is unlikely that anyone will be able to just come up with a plan now that will address everything which is going to happen as we move forward. This is an evolving situation. But we do need to start addressing it now, because if we need to collect additional data, or if we need to trim the data that we're collecting, we need to implement that from now, rather than waiting at the end and then try and, hope that your statistician can pull the rabbit out of the hat and address it then. So as we update things, it's very important to have the version control. We may even find you need to change the study design itself and the status. So part of that evolving situation is, are you going to continue to recruit? Are you changing the population characteristics? And if there is this perceived end of a COVID-19 sort of lockdown state, do you revert back to the original protocol criteria? Or do you continue forward with any changes that you've implemented? The difficult part is it is unlikely we will all come up with the same solution. And as we start to see research published, we'll see that different groups have made very different decisions, and it is likely that the same objective may have different results. So an intervention may work under one set of decisions about how to treat the data, what data to collect and what analysis to use, but a similar objective, collecting in a different way and addressing different endpoints, over different time analysis to adjust for COVID-19, could potentially come up with different trial results. So we need to document all of those decisions we're making all the way through. And we need to think about them ahead of time. We need the study periods definitions clearly set out, and this is probably by site. So if you're having multi-research centres involved, you may have different restrictions imposed at different points in time. You need to start recording, for example, when borders open and shut, when individuals are able to mix. I mean, within Australia, we have the clear stages 1, 2, 3, and 4. For your different regions and your different areas, I would start recording those start and stop dates for that now, so it allows you to know when these rolling periods are occurring. You may need to do separate analysis for these different periods of time, or at least show the results are consistent from one stage to another of COVID-19 restrictions. And again, that may differ by site. We may have different dates things are implemented, for example, in Western Australia, as opposed to New South Wales or Victoria. So the imminent actions, I would imagine many of you have already taken, relate to data collection, how the study is conducted. Hopefully many of you have statistical analysis plans, and you're brushing those off now and thinking about how those will change, whether you want to do an interim analysis. I think it's a good idea to consider whether it's necessary to perform an interim analysis at the start of the COVID-19 restrictions. It's good to get that benchmark situation. How were things progressing prior to the COVID-19, the start of the COVID-19 restrictions? We may need to go back and look at sample size or the power, or what I normally call the trial operating characteristics. How have our assumptions changed? Is our power calculation we performed previously, can we still say we still expect the same parameter estimates for all of those moving forward? The communications need to change as well. So who needs to know what and when? So some of the examples are around unblinding. If someone's about to go into ICU, we may have increased rates of unblinding study medications for ongoing trials. and also how we interact with our data safety and monitoring boards if you have one of those. So going through some of the examples of what has been occurring. Okay. Around recruitment, it may have been halted, postponed, suspended, or they may be finding alternative ways of identifying and recruiting individuals, including changes from changes to the consent process, informed consent process as well. One of the things that we immediately looked at in our protocols was if we needed to trim the data. You know, how much of the data that you're collecting is absolutely need to have, as opposed to, you know, quite nice to answer a few extra research hypotheses as you go along. If you're having trouble collecting data or you're anticipating greater amounts of missing data, go back to your research and identify what are the core variables. What would you not have any research to report if you didn't at least have variable X? Yeah? Put your resources where you're going to get the most return. So identify, and usually we have that concept of levels of importance of data within the protocol. There's also the potential for additional safety data and also data for collecting those different periods. So we really want to know if individuals have missing data, why is it missing? And also when we report out that missing data, we need to do it by those different phases in the post-COVID-19 phase as well. Was there a problem in collecting this data even before we started? So start looking at missing data rates now, common reasons for protocol violation, even before the COVID-19 situation started. Within the intervention itself, we may no longer be able to administer the intervention if it involves coming into the hospital. However, with the restrictions lifting now, we may be able to restart some of those recruitment periods. There are other supply chains that could occur as well, or drugs going out of date. There's also around study drug just being unavailable if they can't physically get it into the country for distribution. In some cases, we may be looking at a different delivery mode of the intervention, if it's possible to switch from intravenous to oral, particularly when you're looking at study drugs. More importantly, look at your standard of care. Yeah? There may be changes in the standard care within the control arm as well. So monitor all situations for your interventions. In some circumstances, I've noticed that some research teams have had to completely change their outcome. So, for example, in our cystic fibrosis study, with patients coming into hospital less, we've gone from a hospital machine measuring the FEV1 to home administration in some cases. However, you have all the overheads of how reliable is that source of data reporting. If we collect lots of data, we could take a mean reading, which may improve the accuracy of less specialised equipment, which is available to the patient to use at home as opposed to within a hospital setting. There may be changes in your study population itself or the environment. So I pretty much covered those before, so I'm not going to go into more detail there at the moment. And also the outcome, we can change the definition or permitted windows. So we may go from saying, you know, the outcome has to be recorded at 28 days. We may say the outcome can be recorded between between 3 weeks to 6 weeks to ensure we have fewer amounts of missing data. So we may find that we're opening up study windows or eligibility criteria for the primary outcome to account for the restrictions that we're all living under. One of the biggest things I want to introduce today, and I'm not sure how many people are aware of them, is when we do prospective studies, intercurrent events occur. So I personally think a better definition of this is post-randomization events. And I'm going to take you through some examples of those. And these are really the fundamental ways in which analysis can change from research team to research team, or depending on which statistician you ask for their advice on how to handle the data. So examples of them are things like missing data due to COVID-19 or death, or maybe a drug holiday, use of rescue medication, or a study withdrawal. They're all events that occur post-randomization, and we need a standardized way before we look at the actual data of how we're going to address all those situations in the statistical analysis. I'm going to come back to intercurrent events on the next slide. If we're doing blinded trials, we also need to look at the impact of drug shortages and also safety monitoring. It may be necessary to unblind far more individuals during a highly restrictive period of COVID-19, as opposed to as we start to try and get back to some level of normality in our daily lives, sort of 6 months, a year further down the line. And also adherence and access to the intervention itself. So some of the potential solutions that we've looked at so far have concentrated on data collection, in terms of amending the data collection, identifying the priority of your data. So the idea your data has different levels, trimming it, considering longitudinal data, which is far more robust to missing data, changing your windows for when you collect the primary outcomes, and those types of solution. When you think about the data collection, you also have to think about the environmental data you need to collect as well. So at this site, at what level of restriction, COVID-19 restrictions are in place at this point in time, and finding somewhere on the database to record this sort of information. You may be increasing your data collection in terms of, was the individual at any point recorded as COVID-19 positive? What was the other one? Sorry. So your database itself may be changing. We need to collect more information. And we need a bit more flexibility in our database, because as this is an evolving situation, you may find in another couple of months you may need to add a few more variables to the database as well. So there needs to be some flexibility in database setup. So we've covered really study conduct. The statistical analysis that themselves may no longer be valid. If you have a large number of individuals who stop taking treatment, or if you're doing a survival analysis and a large number of individuals are suddenly censored because the treatment's no longer available, you may be looking for different underlying analysis or sensitivity, performing sensitivity analysis at different time points as well as overall. I'm going to particularly come back to sensitivity analysis on subsequent slides when I introduce the concept of estimands. Please go back and double check your sample size calculations. Make sure that your results will be able to come up with a valid conclusion, and that you're maintaining the integrity of your trial. And also you may want to, if you don't already have a data safety and monitoring board for prospectively collected data, you may want to institute one, Yeah, they will help be an independent group that helps you make decisions as you go along and weigh up external evidence that may impact on your research as you go along. For many of our trials, we've established a greater frequency of our data safety monitoring board meetings, not just to keep an eye on the safety, but also to get independent advice on changes we want to make to inclusion-exclusion criteria, to changes to the intervention if it's necessary, and also for independent review of changes to these statistical analysis. And these come with revisions to that DSMB charter as well, so document what you're doing as you go along. You also may need additional validation. If you've changed, for example, from a face-to-face assessment to an online assessment, or you change the machines which are taking measurements, you need to cross-validate how you're doing it now under the post-COVID-19 restrictions compared to how it was prior to COVID-19. So consistency, consistency, consistency. And documenting these changes is essential. Each protocol will need an update. It will need a section to set each ongoing research project within the protocol. It's hard to say how much of a recommendation to make for this. My personal recommendation is that every protocol should be updated to include a section to verbally explain what the anticipated effect of COVID-19 is on their research project, and how each of these situations are addressed in the changes within the protocol, or a justification of why no changes are needed within the protocol. So sort of a verbal explanation of what your strategy is, how you approach this. In addition to updates potentially for the outcome, the eligible individuals, visit structures, methods of data collection, and all the other things that we've previously discussed. If you don't already produce an analysis plan, this is a really good time to start. Yeah? To think ahead of how am I going to address all these problems. And again, including that section in the analysis plan, just a verbal, a little text section to say, we anticipate these changes, we anticipate these problems. This is the strategy we've taken to address these situations, and then subsequently all the actual changes you need within that analysis plan as well. Now, previously I talked about intercurrent events, and there's a framework called ESTIMANS, which allows you to bring all of these potential changes together and explain what your future analysis is going to be. So intercurrent events can be things such as treatment discontinuation due to an adverse event, toxicity. It could be due to lack of efficacy. For example, if they're taking an antibiotic and there's no response after a couple of days, clinicians will often choose to switch the antibiotic to bring their symptoms under control. Use of rescue medication, completely switching treatments onto a different arm, or a change in the background therapy, or even a change in the standard of care. You can see from here how the different ways that we treat things can affect the analysis. For example, if you just look at the use of rescue medication, this is a common little bugbear that the FDA brings up with in the pharmaceutical industry. The drug company often would like to exclude individuals who, after the randomization, go on to have any form of rescue medication. Yeah, so they get their nice clean population that usually reduces the variability, and they can usually claim a much larger treatment effect by excluding individuals who received any rescue medication. The FDA, on the other hand, are more interested in the more real-world data, and they prefer to see the analysis with the use of rescue medication left in. But as you can see, there's two very different research hypotheses of whether you just study those people who never go on to receive rescue medication, as opposed to those who potentially need rescue medication later on in the trial. So you can see how an intercurrent event and how we treat that can affect the final statistical analysis that's used, how the data's coded, and what the eventual population estimate of effect is. So as we change the eligibility, the coding of the endpoint, the treatment of those intercurrent events, and our choice of summary statistic as well, or statistical analysis sometimes, we change the question that's being asked, i.e. we change the study objective. So similar trials, by doing slightly different things, for example, including those who took rescue medication or excluding them, can have different and potentially conflicting results. To bridge this gap between the study objectives and the statistical methods, a new framework has emerged called Adherence. And there are guidelines, whoops, I forgot to mention on the slide earlier, sorry. Yeah. So these guidelines on Adherence can be found in this research link and this source down here. And there's actually a whole training module on this particular framework for performing clinical trials. And I'm going to briefly take you through what an estimand is. So on the previous slide you saw, we talked about inclusion-exclusion criteria, we talked about what the endpoint was, we talked about treatment of intercurrent events, and how it's reported. Yeah, what our population summary stats is. I would add 6 to this, which is sensitivity analysis. So normally those are the 6 points you try to address in this framework. And you can see that COVID-19 restrictions and how we treat things moving forward fits in very nicely into this framework. So if our population of interest has changed, if we've changed our inclusion-exclusion criteria for eligibility, we can document it under 1. If we've had to make changes to our endpoint or how we're measuring it, it can get documented under point 2. If we're being more flexible in how the treatment is or the intervention is being administered, yeah, we can document that in point 3. All of those intercurrent events, all of that missing data, how it's treated, how we treat treatment holidays, switching of treatments, increased safety reporting, reduction of collection of confounders that may not be able to be collected during this time. So all of that can fit within the intercurrent events here. And finally, hopefully, we have a similar population level summary statistics for the variable. But actually point 5 here may actually mean that your population parameter you use to say what the treatment effect may be broken down into the pre-COVID-19 period, and then under different restriction of COVID-19, you may choose to report those all out separately. You may even just choose to report every 3 months separately in your protocol, because you may find that overall, this is not an excuse to hunt down a significant p-value. It's not saying dice up your data until you magically or by chance hit that less than 0.05. Yeah? What you're looking for here is consistency. If you have an overall treatment effect, did you also see that treatment effect in each period? If you don't have an overall significant treatment effect, did you have one that was suggestive of efficacy pre-COVID-19, and then it ended up swamped by the increased variability in post-COVID-19? So we're looking for consistency across the whole time period, and the idea that in the future, things will be broken down into these time periods, just until this evolving situation stabilises. It'd be great to start documenting that now, yeah, before you look at the data. If you start peeking at the data and start looking at how things are emerging and then start answering these questions, it's going to be very hard to be objective. If you want more information, there's a nice brief summary. Another consulting group called S-Cubed in the UK produced quite a nice summary of this if you don't want to plough through all of the guidelines themselves. I find them a riveting read, but that's statisticians for you, sorry. So I suppose one of the questions is it still valid to do a main analysis ignoring the impact of COVID-19? I think that's unlikely. However, I'm sure it will still be done, but there should be multiple supporting sensitivity analyses that do account this potential impact. And within this Esteman framework, sensitivity analysis can be defined that account for regions, for time-dependent interactions. And it is entirely likely that you will have to keep updating these estimans as the situation evolves. But really, the future really is looking towards time-dependent interactions in our statistical analysis. So some of the suggested strategies that I covered off today, really the idea that as we go forward in time, we have this idea of rolling enrollment periods, cohorts of patients going through under different COVID-19 restrictions and the impacts they may have on your research protocol and the collection of prospective data. Try to benchmark at the very beginning, before any impact of COVID-19. Yeah? Look at the aggregate blinded data and the evolving situation, just to see if your trial can continue or your research can continue to maintain the integrity of that trial, the patient safety, and the likelihood of being able to report an outcome. Some research may have been compromised completely by the COVID-19 health emergency. We need to identify and record individuals that have been affected by COVID-19 in a time-dependent way. You know, when were they impacted? How long was that ongoing? And how they were impacted by COVID-19. And also at each region level as well. How was each region impacted, as there was an absolute minimum, the start and stop dates for various levels of COVID-19 restrictions. We need to document the percentage of missing data in all these sort of periods of time, and the reason for that data being missing as well. One of the sensitivity analyses that may even need, as an extreme, you may need to remove some of those time periods and say that during that period in time, the assessment of the outcome or the delivery of the intervention is so compromised that we cannot use it between certain periods of time. But again, decide up front, don't try and justify it having looked at the data later. You may need to you will need to document changes in access, availability, and adherence to the intervention over time. In addition, you need to document changes in enrollment and the population characteristics over time, as well as protocol violations. So think of these cohorts moving through your study and document each of those to make sure they are comparable. And if your method of data capture or outcome has changed, then you need to validate that as well. and also recognise it may have introduced more variability into your outcome measure, and you may need to go back and look at things such as your sample size calculation. Document those changes to the definition of the analysis populations, any changes to the outcome, and also the analysis methods, including additional sensitivity analysis. And I would strongly recommend you use the Estimans framework. It just puts it into a really nice, clear situation. And that will translate as well when you publish it. So it could be an appendix, or you can make the stats analysis plan available where your Estimans are clearly documented in an appendix or online material. And don't forget, you may require changes to your trial database itself. Okay, so hopefully I've covered off all the key messages from the beginning in terms of existing guidelines looked at participant safety. COVID-19 restrictions are going to be here for we're talking about years, we're not talking about weeks. You may need changes to multiple things within the protocol and the stats analysis plan. They should be documented, there should be version control, and they should be regularly updated as with the evolving situation. And we need to document the rationale for the change itself. Don't just say, this has changed, and this, why did you take that approach? And it's not just our own publications of this research as we move forward, but also how we evaluate others' research in light of the COVID-19 restrictions as well. Why should we trust how they treated their data, how they were able to collect it, their missingness, their change in population definitions? So it's a two-edged sword really coming in there. Thanks for listening today.
Thanks, Julie. So clearly this is really important, and I think for most researchers the penny hasn't dropped yet how important this is. And I would encourage you, any of you here today or offline, to please let your colleagues know about this. This seminar will be recorded and uploaded to our website, and it'll be available for at least the next year online, if not longer. And I think it's really important that we spread the message of why this is critical to think about. And clearly the journals are going to be looking for this, and if we haven't accounted for these factors, it's going to be problematic. Are there any questions today? I couldn't see any online, but anyone have any comments or questions here today? No? Well, thank you very much, Julie. I thought that was very comprehensive, and I think it's really important food for thought. And obviously this affects everyone doing research, so we really need to get that message out there to everyone. So thank you for attending today, and I'm sure Julie will stay behind if you think of something on your way out. And otherwise, please spread the word about the recording being available downstream. Thanks again for coming. And please thank Julie. And please fill in your evaluation form if you get a moment too. That would be fantastic.